Now let's move on to the membranous organelles. The first type of membranous organelle is the mitochondria. Mitochondria have a double membrane structure. There is an outer membrane and an inner membrane that folds to create the cristae. Inside the mitochondria is the mitochondrial matrix. It has fluid and metabolic enzymes. The function of the mitochondria is to produce energy. They produce ATP to be used by other cellular structures. 95% of the ATP produced by your cells is produced by the mitochondria. If the mitochondria become dysfunctional, as is what happens with poisoning by cyanide, death occurs quickly because cells cannot produce energy to sustain life without mitochondria. The rough and smooth endoplasmic reticulum is a network of membrane-bound sacs and tubes. For a rough endoplasmic reticulum, the surface is coated with ribosomes. Because ribosomes are involved in creating proteins, the ER is responsible for changing and packaging proteins for export to the Golgi apparatus. As the protein is manufactured by the ribosomes, it is put into the endoplasmic reticulum. The ER will fold and change the protein before sending it to the Golgi. The smooth endoplasmic reticulum lacks ribosomes and is much more tubular. This type of ER is involved in the synthesis of lipids and steroid hormone. The smooth ER is also involved in glycogen and calcium storage. Next up, we have the Golgi apparatus. The Golgi apparatus consists of a stack of hollow sacs, like a stack of pancakes. These stacks are called cisternae. The function of the Golgi apparatus is much like a shipping and receiving department for the cell. It receives newly modified proteins from the ER. It sorts those proteins and may change them further. It then packages them up into vesicles that are sent throughout the cell, or they can exit the cell through exocytosis. The lysosomes are small spherical vesicles that have digestive enzymes to break down large macromolecules within the cell. They also destroy any bacteria that enters the cell. The peroxisome is a very small spherical vesicle that absorbs and breaks down fatty acids and other organic compounds. They also help to neutralize hydrogen peroxide formed by fatty acid breakdown. Peroxisomes serve to protect the cell by preventing reactive oxygen species from damaging important cellular proteins and DNA.